All right, so today we're going to continue the lecture on genome annotation. I was telling you about what are genes, and you're going to use a bioinformatics tool to predict genes. And I gave you an exercise where I gave you a sequence of a part of a chromosome, and I was asking you, where do you think the genes are? Because I want you to know that one approach of predicting genes, which is called ab initio approach, which is like from the beginning. And it's looking for those cues of where is a possible start codon and where is a possible stop codon. Are they in frame? And in frame means when I each every 3, when this is a start codon, the ATG, then over here, is this stop codon still these 3 letters being read, or does it shift around and therefore not a stop codon? So gene predictor softwares that are looking at this sequence and trying to predict where a gene is, they're looking for the start codon, the stop codon, which are the 3 nucleotide sequences that indicate beginning of translation, not transcription, beginning of translation, end of translation. But there are parts of the gene that actually, there is a part that is transcribed, not translated. So there is a chunk on the 5' end that's before the start of translation. So before that start codon, that is actually part of the mRNA. Let's see what else. Oh, we were talking about Tata Box. And I put some links there in case you are curious about how far up it has to be, upstream from the start codon. You don't have to memorize the numbers. I just wanted you to know it has to be in the vicinity upstream, and that's something that the software also uses to predict where a gene is. All right, so let's see where we ended up, and you can ask me questions if you're like, okay, this part I'm still not comfortable with. This part we already went over, so I'm gonna go a little fast. I think this is, no, I think I should go back to this in a little bit. This might have been where we ended. Yeah, so in this question I was asking, could this ATG base pair 56 and 59, which is over 56, okay, over here, this one, could it be a start codon, and then this one would be the stop codon? And the answer to that is no, for 2 reasons. If I read every 3 starting here, then this TAA wouldn't be read as a TAA. So let's see. ATG, the first codon, ACT, the second, 3, 4, 5. Yeah, so the codon here would be ATA. Therefore, TAA doesn't get read as a TAA because ATA is one codon and then ACG is another codon. So this TAA is out of frame to be a stop codon. The other thing is that let's pretend that this was in frame, but this ATG is very close to this stop codon here. And that would make a protein that is so small. It's not even a peptide. It's way too small to even be anything. So how do you know it's too small? You look down here. This is the what fraction of the proteins of some organism. I don't remember which organism this is, but what fraction of the protein is how long. And then you can see there are proteins of different lengths, some pretty big proteins, but there's a minority. And then most of them are, I don't know, 200 to maybe 250 amino acids. And then you're like, oh, okay, amino acid, but for each one amino acid, there are 3 nucleotides. So you could say, oh, okay, then each, let's say here, a protein that is 250 amino acids, then it would have at least 750 nucleotides, because it has to be 3 times that many. And this is for you to conclude that that ETG is way too close to this TAA for this to be a gene. So a gene predictor software that is looking for cues, it looks for start codons, stop codons. It also looks for Tata boxes. So it has to be a number of nucleotides upstream of, let's see, there's this one, and then there's this one. So then the gene predictor software, you will see, is it this one? Where does this gene start? Is it here or is it here? And then it counts how many nucleotides upstream is that Tata box. So this Tata box here is something that the RNA polymerase uses to know where to bind. So imagine that this is some DNA fragment, and that DNA fragment becomes an mRNA or an RNA molecule. Maybe this is the gene for ribosome. So there is a DNA version which gets transcribed into an RNA version by an enzyme called RNA polymerase. So the RNA polymerase will bind this, and you will read this scanning for it actually will transcribe this part, and this is going to end up as part of the RNA. So, okay, those are cues that the gene predictor software uses. They're not all of them, but they're the ones that are easy for me to show you. So here I'm actually just giving you the distance, but I just want to, so that you have some idea of where is the Tata box. And when you use the gene predictor software, you're going to see there's something called TSS, which is the transcription start site. And I'm telling you that's not the translation start site. The translation start site is the start codon, the ETG. The TSS is the transcription start site, which is different. And so this is the word I was saying when I said ab initio from the beginning. And then the software is looking for those cues. There is another approach that's called homology-based. And what would happen is that we give BLAST this sequence, and we say, BLAST, if this was translated to protein, which protein would it be? And since BLAST doesn't know if this is the template strand or if it's the coding strand, then it will translate this in 3 frames, reverse transcribe it, or reverse complement it, and then translate the reverse complement of this, make 6 proteins, and then check if each of these 6 proteins match a protein in the database. So that's called the homology-based approach, which you are going to do. So if you don't understand this bit, don't worry about it. Mainly, I want you to know that there are at least 2 different ways of predicting genes. One is just looking for the cues within the sequence, and the other ones to say, is there a protein that comes out of any of this sequence that we know of? So good? So far, so good? All right, thank you. All right, so here's some representation of what we could use to predict eukaryotic genes. Okay, I told you about let's see, the start codon. So this transcription start site, and over here, or maybe over here, there would be like the start codon would be the beginning of the exon 1. So it would actually be right here. I don't know, why did they put TSS? If this is, yeah, sometimes there is some space between the transcription start site and the start codon. That's actually most of the cases. This figure doesn't display that. But imagine this is a DNA sequence, so DNA fragment, and there is a part upstream of it that does not get transcribed. It's not entirely true, but let's say the promoter does not get transcribed. This is a small part that gets transcribed, not translated. But this blue chunk here is where the machinery that will do the transcription will bind. So in here, there's a data box I told you. There's something called a cat box. So anyway, there are signals in here in the promoter that tell the cell to transcribe the stuff, the sequence that is downstream from it. And then gene will have, not every gene, but some genes will have exons and introns. For eukaryotes, prokaryotes don't have introns. So what the software can also use is like these cues. These things here are the intron-exon boundaries. They can also use that to see, oh, where might the introns be? And let's see what else. And mostly that. And here's, this is stop codon. Weird, the stop codon is ending before end of exon 3. I would put the stop codon at the end of exon 3. This part here would be a part that, so there was DNA version of it. There is the RNA version of it, which doesn't end up in the protein. So this chunk here gets removed. And so this is the 3' UTR. That's the 3' untranslated region. So it's a part of the DNA that gets transcribed, but does not get translated. See how this chunk is missing from here? So instead of translating this part gets cut off, and then the cell puts a bunch of A's here. It's called poly-A tail. And then the cell will read the exons. The ribosome is going to read the exons. It will not translate the 5' UTR. And the parts that it reads and puts the corresponding amino acid, that's what form your protein. So the next slide is about comparing the two different approaches, but I already told you about all of this. So there's the ab initio prediction, and there's the homology-based. We are going to compare DNA sequence using BLAST, and what we're looking for is, are there these matches to the DNA sequence? And the ab initio are those cues that the program is using. And as you might imagine, predicting genes is a big deal in annotating genome. That's why I'm spending so much time in it. Also, you know, it's kind of important that you know what is a gene, the physical aspect of it. So I always ask that in exam. For you to define the gene, I don't want you to Google it, because if you Google it and just do a copy and paste of what a gene is, then I can generally tell there was a copy and paste. I want you to say it in your own words. Okay, so I'd like you to be able to tell the differences between prokaryotic genes and eukaryotic genes, and the difference between genomes. So the next slide, I'm going to show it to you, and I want you to find the 7 differences, or I don't know if there are 7, but I'd like you to compare them. I'll be quiet for a moment, and you find what's the difference between this one is the prokaryotic gene, and this is an eukaryotic gene.